Hello, I'm Priyanka, Senior Bioinformatician at the Wellcome Sanger Institute in the UK. Today, I'm going to talk about our automated pipeline to generate and submit genome notes. The Darwin Tree of Life project is a collaboration between biodiversity, genomics, and analysis partners from 10 different organizations in the UK, institutions, museums, and universities, with the aim to capture the diversity of life on these islands. The Darwin Tree of Life is just one of several initiatives working towards the goal to sequence all complex life on Earth. This should help in conservation, to understand the interconnectedness of all life, and to build a new economy based on biological materials. We use genomic data to understand this diversity of life. And with that aim, we are targeting over 70,000 different sequences across animals, plants, fungi, and protists. The life on the islands of Britain Island is diverse and contain members of over 40% organism families. In the first phase of the project, we are targeting sequencing 2,000 species over 2 years, and then the project will scale up to sequence over 70,000 species within 10 years. On the website on the right, GOAT, which stands for Genome on a Tree, several of these biodiversity projects are tracked and show what sequencing targets have been met. Currently, for the Darwin Tree of Life, we have sequenced over a quarter of the phyla that we were targeting, and more than 5% of the families that we aim to study. At the bottom, the pie charts show the status of the samples from when they are collected out in the field to when they arrive here at Sanger and once they are in sequencing. Once sequencing is done, then the raw data goes through a series of production-grade automated bioinformatics pipelines designed with Nextflow, which is a workflow manager, and using NF code template, which was designed by a community initiatives, which makes all our pipelines easily reproducible and relatable. With all our pipelines, we do continuous testing using diverse size taxon and data sets, starting with the really one of our smallest genomes, the chicken of the woods, which is 38 megabases, to one of our largest published genomes, the red deer, which is at 3 GB. Currently, we're also working on the European mistletoe, which is a 90 GB genome. All this is deployed for production in-house using our in-house server, using a database and WestKit. In all aspects of our work, we try to embrace the principles of open science, transparency. All our tools and pipelines are built on GitHub, where we welcome contributions, comments, and critique from the development community. Our datasets and downstream analysis results are released without embargo. All our pipelines are extremely robust, thanks to the testing that we do against small, medium, and large genomes from different taxa groups. Our resources are assigned to these pipelines according to various input size statistics to make sure optimal utilization is achieved. All our pipelines are also reproducible, and every release is documented both on GitHub and Zenodo, another open source database. The pipelines also use only one container per process, which makes the installation trivial, the updates easy, and the results highly reproducible. A snapshot of the production engine that we are designing starts with assembly stage, then moves on to the curation, followed by contamination removal once the data is uploaded to ENA and passed on to NCBI. Our partners at Ensemble carry out some of our repeat and genome annotation work. All of this is then passed on to the final stage, which is nicknamed Genome After Party. Genome After Party, the main goal is to generate these genome notes. Genome notes are our automated publications that allow us to share data, code, and results with the scientific community and the greater public. They summarize the key methods, the results, and the results of each genome, which are generated using a series of automated pipelines, starting with downloading the genome from NCBI, uh, collecting the results from read mapping, variant calling, and blob toolkit pipeline, calculating contact map matrices and genome statistics. As you can see from the structure, the pipelines are highly modular, with one container being used per process, making the installations trivial. And also, this allows us to share different aspects of our pipeline if needed. All our data sets and results are released immediately without embargo. Finally, we are also working to integrate this pipeline into a platform which will automatically gather results for any species, write the genome note, and then submit it directly to the publisher. Here is a preview of our Welcome Open Research Gateway, where all of these genome notes are published. And on the right, you can see an example of the gray wolf genome note. Thank you. I would especially like to thank the Slack community for Nextflow and NFCore, which have helped us with technical questions during development, and all the informatics team members in Tree of Life program here at Sanger, who have helped me extremely with discussions and their feedback. Thank you.